Okay. All right, so I'm recording now the session. You guys should be able to see a little camera with a red dot in it. Is that correct? Yes? Just tell me whether you could see the little camera with the red dot in. Good, okay. All right, so let's start over here. And this is lesson 2 on chapter 12. All right. And before we go a little bit forward with material, I want to review something. I want to review some important examples of ion channels, right? And I'm particularly now interested maybe in the gated ion channels, to talk about examples of gated ion channels. But before I mention the 3 major categories of gated ion channels, I do want to mention this idea of leaky channels, okay? And leaky channels also exist for different ions. And the most famous leaky channel is the potassium leaky channel, which is found in the plasma membrane of animal cells, such as our cells, correct? And this is the most famous one because it plays a role in maintaining the resting membrane potential, okay, that all living cells would have. So if you're a living cell, you're going to have a resting membrane potential across your plasma membrane. That's something that cells do, right? And this idea of the potassium leaky channel means that it's not gated. There is no trigger that tries to open it or close it. It periodically goes through different transitions of opening and closing, random opening and closing. And when it's open, it allows whatever ion it's geared for, which in this case would be potassium, to passively move into the cell or out of the cell, depending upon whatever the situation of that cell is. Because remember, channels are always, whether they're gated or not gated, they always involve passive movement. Passive movement of whatever ion that they are characteristic for. All right, so the most famous leaky channel is the potassium channel in the plasma membrane of animal cells. All right, so just keep that in mind. And that's not a gated channel. Now, when we talk about gated channels, we talk about voltage gated channels. And here we mentioned 3 important voltage gated channels. All right. The voltage gated sodium channel, that's in the plasma membrane of the nerve cell axon, associated with action potentials. Some of you may have already taken like neurobiology, so you should be really good with a lot of the aspects of this chapter if you already took neurobiology, right? Then we also have the voltage gated potassium channels, which again are found in the plasma membrane of the nerve cell axon, that is responsible for bringing the membrane potential back to resting potential after an action potential is moving through, correct? All right, so those 2 channels that are present in the axon, right, okay, of nerves are very important to know. And then we have this other important gated channel that's very important to know, and that's the voltage gated calcium channel, which is found at the end of the neuron, at the nerve terminal, okay? And the voltage gated calcium channel, when the action potential gets to the end of the nerve terminal, the action potential, instead of opening up a sodium channel, opens up a calcium channel. And this calcium channel is very important at the nerve terminal for stimulating the release of a neurotransmitter, okay, at the nerve terminal. All right, calcium channel. So these are 3 very famous voltage gated channels. Okay, and then we also have quite a number of different ligand gated channels. And a ligand will refer to a particular chemical. We're not talking about a change in mem potential. We're talking about a particular chemical that will be released, that is generally a neurotransmitter that will be released, that turns around and can bind to an ion channel and increase the rate of it being opened. So, of course, we have the famous acetylcholine receptor, okay, which is really an acetylcholine gated sodium channel, cation channel, okay, that's found in the neuromuscular junction, right? So in other words, when the nerve terminal actually releases its neurotransmitter, the neurotransmitter in the neuromuscular junction is something called acetylcholine, ACH, right? And of course, this is an excitatory neurotransmitter, which means a neurotransmitter that actually binds to its receptor, the receptor that is in charge of the channel. Okay, if it's excitatory, it's going to end up causing what? An action potential in whatever the excitatory tissue is, whether or not that's a muscle tissue or whether or not that's a nerve tissue, or sometimes it could even be an endocrine tissue. All right, so the acetylcholine receptor, which is a what? Which is a gated sodium channel, which is in charge of. Then we also have some other ligand-gated channels, such as the glutamate receptors. And glutamate, and remember, glutamate is an amino acid. It's an amino acid. Glutamate's an amino acid, but here it's not functioning as a what? As a building block of a protein. Glutamate is in its own right a very important neurotransmitter. In fact, it's an excitatory neurotransmitter, okay, in the nervous system. So glutamate receptors, okay, actually also open up sodium channels and cause depolarization at the synapse, okay, and is considered to be an excitatory neurotransmitter. Okay, acetylcholine is also an excitatory neurotransmitter, but really that one is more famous in the neuromuscular junction. In the nervous system in general, okay, glutamate is a very famous excitatory neurotransmitter that causes sodium channels to open up in the synapse, okay, and stimulate an action potential in the postsynaptic membrane. Then opposite glutamate is GABA. And interestingly, gamma aminobutyric acid is actually made from glutamate. So the precursor to GABA, like where does GABA come from? GABA is actually made from glutamate. And it's so interesting that GABA, okay, is an inhibitory neurotransmitter. Okay, so an inhibitory transmitter is not trying to generate an action potential in the postsynaptic cell. Okay, an inhibitory neurotransmitter is trying to quiet down the cell instead of exciting the cells. And I always found it very interesting that GABA is actually made from glutamate. So glutamate, one of the most famous excitatory neurotransmitters, okay, and the nervous system, is the precursor to GABA, probably the most famous inhibitory neurotransmitter. All right, so excitatory neurotransmitters, when they end up binding to their receptor, they usually cause depolarization of the membrane and excites the membrane, maybe brings it to threshold, and an action potential is initiated. Whereas instead, if GABA is released, GABA is an inhibitory neurotransmitter that binds to the GABA receptors, and GABA receptors actually prevent depolarization and prevents the postsynaptic membrane from moving to threshold and generating an action potential. All right, so we want to know glutamate is an excitatory neurotransmitter. Got it? We want to know that GABA, okay, is an inhibitory neurotransmitter. And the other inhibitory neurotransmitter that's famous in the nervous system is another amino acid. And, you know, for me, especially as a cell biologist, biochemistry person, I really consider it interesting that we only think about amino acids being important in protein structure, but many amino acids have their own separate function and role that they can play, okay, in cell biology. So glycine, which is another amino acid, right? Glycine is also another inhibitory neurotransmitter in the central nervous system. And we'll explain a little bit later how they act as inhibitory neurotransmitters, but here it gives you that information. Instead of opening up a sodium channel, notice what the inhibitory neurotransmitters are doing. They're opening up a chloride channel. So we'll come back to that a little bit later when we start getting deeper into actually how these things work in the rest of the chapter. All right, so if you're an excitatory neurotransmitter, you're generally going to be opening up a sodium channel because a sodium channel will cause depolarization. right? And depolarization, if you reach threshold, enough depolarization reach threshold, you'll set off a new action potential. And obviously, you'll continue the electrical signal, okay, onto the next neuron by doing that. On the other hand, if you want to quiet the next neuron and you don't want it to fire, okay, you would use an inhibitory neurotransmitter, and that doesn't open a sodium channel. Okay, what it opens instead is a chloride channel, which has an opposite charge, and chloride will run into the cell and will actually try to prevent depolarization and prevent the postsynaptic membrane from reaching threshold. Okay. All right. So therefore, there's a number of different ligand channels that are here that are famous. Now, these are not the only ones that exist, but these are the most important ones for this level of cell biology and the aspect of neurobiology that we're talking about in this chapter. And then, of course, the third type of gated channel are the mechanically activated channels, the stress activated channels, which we find in the auditory hair cells in the inner ear, the organ of Corti, which is involved in detecting sound vibrations, which we talked about in the first lesson, and we even looked at the little video, okay? And that's really the most famous mechanically activated or stress activated channel. Then again, it's a cation channel that's going to be opening up sodium ions, and sodium ions, when they rush into a cell, will cause depolarization, and that's why they would be excitatory. Okay. All right, so I just want to review that. So we want to make sure that we are aware of these important gated channels, ion channels, the voltage gated channels, these ligand gated channels, and of course the one and only stress activated or mechanically activated gated channel. All right. Okay, so taking a look here a little more like voltage gated calcium channels. Okay, because we did talk a little bit about the sodium channels in the previous lesson, and we did talk about the mechanically gated channels. All right, so here we are looking now at a resting nerve terminal, okay? But when a resting nerve terminal, okay, which has its voltage-gated calcium channels closed, okay, when a synapse is going to come through, or not a synapse, when an action potential is going to come through, the change in membrane potential when you get to the nerve terminal, okay, the action potential is going to open up a voltage-gated calcium channel. So all during the action potential, we've been opening up sodium, voltage-gated sodium channels, correct? That's what we've been opening up, and voltage-gated potassium channels to do the action potential. But by the time the action potential gets down to the nerve terminal, what we're opening up now is something a little bit different, a voltage gated, and that's because of the action potential, voltage, right? Gated, and it will be a calcium channel. Okay, and when that happens, okay, when the action potential comes down, right, the action potential, okay, reaches the nerve terminal, okay, that change in voltage of the membrane potential is going to now open up a calcium channel. And when you open up a calcium channel, all right, the calcium is going to flow into the cytosol, right? And cytosolic calcium is a super important trigger. It's a real cytosolic triggering structure, okay, that actually does a lot of different things depending upon what the cell is. So when calcium is suddenly released into the cytosol, it stimulates the synaptic vesicles with their neurotransmitters to now move to the plasma membrane, and through exocytosis release the neurotransmitter. Okay, so here we have an electrical signal coming down. This is electrical, right? This is electrical signal. And this electrical signal is now turning into a chemical signal because we're now getting a release of the neurotransmitter, whatever that is. So this could be like a nerve ending and maybe in the neuromuscular junction, right? So at the end of the nerve terminal, okay, the neuromuscular junction, you're going to have the action potential opening up calcium channels. The calcium is going to cause the synaptic vesicles with maybe the acetylcholine that's in there to now fuse with the plasma membrane and release the neurotransmitter. Maybe this is ACH if this is a neuromuscular junction. Okay, and that actually means the electrical signal has been changed into a chemical signal, right? And this chemical signal is now going to go ahead and find the acetylcholine receptor that's going to be in the postsynaptic membrane, right? Because this is the postsynaptic cell. And there's going to be a receptor there for the neurotransmitter. And in the neuromuscular junction, that's going to be acetylcholine. And of course, that means that these little guys are going to open up. These little guys are going to bind to what? Its receptor and open up this channel. And if it was the neuromuscular junction, when you open that up, it's going to send sodium in. Okay. And then what's going to happen is you're going to depolarize here in the postsynaptic membrane. Okay, and excite that cell, whether that cell is actually a neuron or whether that cell is another type of excitable tissue like skeletal muscle. Okay, you're going to generate a what? You're going to depolarize the threshold and generate an action potential in the postsynaptic membrane. And so we're talking about on how we have a voltage-gated calcium channel, okay, ultimately changing an electrical signal into a chemical signal, and then that chemical signal is going to interact with a ligand-gated channel, okay, in the postsynaptic membrane. All right. How do we study ion channels? Okay, how do we study that in research? How do we learn about how are ion channels controlled? How are they regulated? What's influencing them? Okay. What is their rate of being open versus closed? What kind of factors can influence that? One of the most famous ways that we study ion channels in cell biology is by doing what's called patch clamp recording. So patch clamp recording, we don't have to go into gory detail about it. We just have to know that patch clamp recording is still one of the most famous ways that we do research to study some of the features of ion channels that are found in membranes. All right, so ion movements across membrane can be detected by electrical measurements, and because of the technical advances that we now have, we can permit the measurement of an electrical current through a single channel molecule by using patch clamp recording. And it's quite amazing because what they do is that they take a glass microelectrode conducting fluid tightly sealed around an ion channel membrane. They literally can pinch off a piece of what? Of membrane material that might include, okay, a channel or a couple of channels, right? And it's cell attached, and then you detach the patch, and then you turn around and you can put that little piece of membrane that is sealed now in this glass microelectrode with the conducting fluid in it. You can actually take off a tiny little piece of the membrane, capture a little channel of interest in that membrane that you're looking at, and then you can actually trace by actually having all this electrical conductivity going on between the what? Between the fluid that you're putting it in on one side and the conducting fluid on the other side. You can actually find out what's going to cause this little channel to actually open and close. Okay, so you can trace an ion channel's currents. Okay, and you can control the fluid that's here in the dark blue, and you can control what's in the fluid that's in the light blue, and you could start to even recognize, well, what kind of ion does this channel even allow to go through, and how does this change the voltage? Okay, how does this change the currents or the voltage when those ions are available? So using the patch clamp recording, you could use it to study ion channels, a large variety of cell types. By changing the ionic composition of the bathing solutions, you can establish which ions can go through the channels that are present in that membrane. Okay, you can clamp the membrane potential at different voltages and establish the effects on channel activity. If the patch is sufficiently small, if you can grab a small, tiny little piece of membrane, okay, a single channel molecule can be captured, and you can measure the ion flow through a single channel. And of course, you're doing it like in picoamps. It's like amazingly small. Picoamps would be 10 to the minus 12 amps. Okay, because we're talking about like a single channel. Okay, current randomly will switch between the two levels due to channels even being opened or closed, and you can trace that. You can trace and actually see, okay, how the voltage reading is going to change when the channel is open versus when the channel is closed. Okay, and of course you could also turn around and see what's going to regulate. Okay, what kind of chemicals or what kind of conditions is going to regulate whether or not the channel is open or closed, or whether or not there could be factors to keep the channel open or closed for a longer period of time. So what they would even have in ion channel recording, okay, you could actually even study the state of the channel. You can see what the change in voltage is going to be when you're recording it, okay, and you can end up adding different factors that's going to increase or decrease, okay, the amount of time it spends open or closed. Keep in mind with all channels, can't spell that, all channel proteins, because we always get this tricked on a test, all channel proteins, well, almost all of them, certainly if they are ion, or ion channel proteins, okay, they always open and close. They never stay permanently open. They never stay permanently closed. They have random protein conformations where they are constantly opening and closing. Okay, if they're a gated ion channel, you can influence, okay, the amount of time it spends open by exposing it to whatever trigger it is. But it's always going to be opening and closing. Okay, a gated channel will a trigger will influence the amount of time it spends open, change the amount of time it spends open. But it's not a point that channels are always open or closed. They're always fluctuating back and forth. They're also never like half open. You never say that you have a channel half open. You're either open or you're not open. Then you're open, then you're not open. You're never going to be partly open. All right. So keep that in mind, because a lot of students kind of mess up on that. They think that a channel always stays tightly closed until the trigger is there, and that's not true. Okay? All right. All right, so let's see. So let's leave the ion channel things out. So let's go back and talk about the voltage gated channels again. Okay? They're the most popular gated channels, the voltage gated channels. That's what we have probably the most of in our body. They play a major role in propagating electrical signals in both nerve tissue and also muscle tissue, nerve cells and muscle cells. Okay. The channels have to have a special charge protein domain called the voltage sensor. So if you had to say, well, what is the voltage sensor on a voltage gated channel? It's a special protein domain, protein area, because remember, these proteins that make up channels, okay, they could be like multi-pass proteins, okay, multi-pass proteins that have different protein locations or protein areas. And so there has to be a specially charged protein domain which is sensitive to a change in the membrane potential of the membrane that it's in. All right? When the potential changes beyond what we call threshold, right? Threshold value, the channel switches from the closed to the open configuration. So it doesn't always tightly close. There'll be sometimes when those channels, even though they're gated channels, they will periodically open. But suddenly, if you actually have a threshold voltage, an electrical signal causes that very voltage sensors, okay, of the channel to actually reach threshold. Suddenly now, instead of it what? Instead of it being mostly closed and only periodically opening up, like just randomly, suddenly now it will definitely open up and stay opened up for a longer period of time. All right. Open state probability increases at threshold potential. See? So open state probability, how long it's going to stay open, is going to increase when you are at threshold potential. All right. What controls membrane potential? So this is an interesting thing here. We say, well, what controls the membrane potential? Why do we even have a membrane potential? What is influencing controlling membrane potential? The opening and closing of ion channels. Okay. But the opening and closing of ion channels can change the membrane potential. Do you agree? All right. So what controls membrane potential? It's going to be opening and closing of various ion channels, right? Okay, and then of course we turn around and say this generates a very important control loop in electrical signaling, that ion channels, okay, are responsible for generating the membrane potential, but then the membrane potential or changes in the membrane potential will affect the opening and closing of ion channels. And this is a very important fundamental principle of what's called electrical signaling in cells. All right. So therefore, you wouldn't really have a membrane potential, okay, unless you had what? Ion channels doing certain things. So ion channels are responsible for causing the membrane potential to even be there. But changes in membrane potential can then in turn influence what? Whether the ion channels, whether the various ion channels are opening and closing. So this is this interesting control loop between ion channels and membrane potential. It's a circular loop. You get it? And this is the principle, the fundamental principle between affecting what's called electrical signaling in various types of cells. All right. The ion concentration inside and outside of a cell is very different. So if we're talking about our typical human cells, if we want to say, you know, what would it be mostly for us? Because again, the book does focus a little bit more on what the human numbers are. We certainly know that we have the intracellular fluid and we have the extracellular fluid. And we know the major cation, sodium, is the major cation of the what? Of the extracellular fluid. That there's a lot more sodium outside the cell than inside the cell, correct? And the opposite would be true for potassium. Potassium is the major cation, okay, of the what? Of the intracellular fluid, that there's a lot more potassium inside the cell than there is outside the cell, correct? All right. Then when it comes to anions, obviously there has to be anions outside the cell, and the major anion outside the cell is going to be the chloride ion. It's the major anion outside the cell, right? Major ion outside the cell. And we have some chlorine on the inside the cell, but you could see a dramatic difference in concentration, correct? And then if we said, who are the major anions inside the cell, we say there are fixed ions inside the cell. Fixed ions, and people don't usually know what that means. Fixed ions mean things that we don't really have, like a channel protein to actually allow a fixed anion to get across. So for example, the main fixed anions that are inside the cell usually make up mostly the phosphate ions, and also charged organic chemicals, biochemicals. So we have a lot more of these. In other words, many of the amino acids that will be inside the cell, okay, at higher concentration than outside the cell, are going to carry a negative charge. Most proteins carry a negative charge, And these are considered to be fixed anions inside the cell. So think about even pyruvate and lactate, and we think about succinate, we think about all of those different players in metabolism that we discussed in chapter 13. Okay, a lot of these are actually negatively charged, right? They're weak acids, and so therefore they still carry what? Negative charges. So lactic acid, pyruvic acid, succinic acid, okay, alpha ketoglutarate, all of these different players, these are considered to be part of the fixed anions, that they don't travel through any ion channels. Okay, even phosphate is too big to pass through a typical what? Ion channel that would be in the plasma membrane. So the fixed anions, okay, that are going to be inside the cell are mostly your organic biochemicals that carry a negative charge, that are in greater concentration inside the cell than outside the cell. Okay, all right, so those are the fixed anions. So a lot of times they say that, nobody knows what it is. All right, so the fixed anions are going to be things that are negatively charged, and they're inside the cell at a higher concentration than outside the cell. Okay, and a lot of them are negatively charged. Like I said, all of those things that we were talking about in chapter 13 with what, glycolysis and Krebs cycle. If you really take a look at all of those chemicals, they all carry what, carboxylic acid groups on them that carry a negative charge. And even like I said, most proteins under physiological conditions carry a net negative charge. And you have way more proteins inside of a cell than you have outside the cell. Okay, so we know this would be an easy question, right? The major cation of the extracellular fluid, the major anion of the extracellular fluid. And who is the major cation of the intracellular fluid? Potassium. And who's the major anion? Major anion is going to be, like I said, all of those what? Those biochemicals, okay, that carry the extra carboxylic acid groups and carry a negative charge. All right, those are going to represent the fixed anions that are inside the cell. All right, we also have a few other ions that have somewhat of a difference. We have a difference in the bicarbonate ion, with more bicarbonate being a little more outside the cell than inside the cell, but there's not a super dramatic difference here. Do you agree? Compared to what we're looking at over here above, right? All right, so there's going to be a little more bicarbonate outside the cell than there's going to be inside the cell. Okay, magnesium, okay, there's going to be what? A little more outside the cell than inside the cell. But one of the most interesting things, okay, even though it looks like we don't have a lot of calcium at all, there's a tremendous difference between the amount of calcium that's going to be free inside the cell versus the amount of calcium that's going to be present outside the cell. So remember, when we say intracellular fluid, this is the cytosol. The cytosol. We're not talking about inside the mitochondrial matrix. This is really these numbers that we're giving here are cytosolic values. Okay, not like in the lumen of the ER, not in the nucleoplasm of the nucleus. There could be different concentrations. These are all cytosolic concentrations. And a slight difference in pH. We usually say the pH of the blood is around 7.4, but the pH of the cytosol is not 7.4. The pH of the cytosol is usually somewhere between 7.0 and 7.2. All right? So that means that there's more hydrogen ions where? There's more hydrogen ions inside the cell, right? At any given time, we would expect more hydrogen ions inside the cell in the cytosol than what you would expect to see in the extracellular fluid. And that's not a big surprise because, let's be honest, metabolism in general is acid producing, and metabolism is really something that is going on inside the cell. So just running your metabolism, just doing your glycolysis and doing your Krebs cycle and doing your oxidative phosphorylation is more acid generating anyway. So the acid is really generated inside the cell. And then, of course, we try to make sure that we don't get too much acid inside the cell. We would send excess acid or acidic things outside the cell, but it's always being generated inside the cell. All right. Ion distribution across the plasma membrane. Here we go again. And remember, intracellular fluid is the cytosol specifically. Okay. Ionic species are not distributed equally on both sides of the membrane, as we just said. Here we got in the extracellular fluid, we got mostly sodium and chloride outside, correct? Whereas on the intracellular fluid, we have mostly potassium inside, and of course, organic anions, as we said, those fixed anions. So higher concentration of potassium and organic anions, like organic acids and proteins, on the inside. Outside is mostly sodium and chloride. The overall effect of this unequal distribution of ions is actually referred to as a membrane potential, an unequal distribution of charges across the plasma membrane. Okay. Okay, a resting membrane potential. All living cells maintain a resting membrane potential. All right. An unequal distribution of charges across the plasma membrane, in so much that it's always positive on the outside of the membrane compared to being negative on the inside of the membrane. Positive on the outside. And again, this is not, it doesn't mean it's all positive out here, you know, and it doesn't mean like all negative throughout the entire, it's really right across this barrier, okay, is where we see a separation of charges. Right across the barrier of the plasma membrane is where the membrane potential is. It isn't far away from the plasma membrane, like way out here. That's not what we're talking about. We're talking about right across the plasma membrane. There's going to be a separation of charges. And what would this positive and negative charge want to do to each other? They would want to meet and cancel each other out, correct? But they can't meet and cancel each other out because why? Because the plasma membrane is a barrier. All right. So the resting membrane potential of a cell, and all cells have a resting membrane potential, is due to an unequal distribution of charges across the plasma membrane. All right. And living cells actually have to spend energy to maintain this resting membrane potential. The resting membrane potential, okay, in voltage, right, millivolts, right, usually millivolts, is known to range between minus 20 millivolts to minus 200 millivolts, depending upon the type of cell membrane, okay? And they always say it's a negative reading, not because it's negative voltage. The membrane potential, the sign that they give you for the membrane potential, whatever is considered to be the charge of the inner compartment, that they use here. So because the cytosolic side of the membrane is negative compared to the what? The outside of the plasma membrane, that's why it's negative 20 millivolts to negative 200 millivolts. It's always negative on the inside compartment of the membrane, okay, and relatively positive on the outside. So this was an arbitrary thing, I guess, that the physiology people decided that we're always going to talk about which side the charge, we want to know what the charge of the inside compartment is. And if the charge of the inside compartment is negative, you say negative 200 millivolts or negative 70 millivolts. It's not a negative voltage reading. It's not a negative. You can't have negative voltage. When you test the battery, you either have positive voltage, you either have voltage, or you're zero. Okay, you're never negative voltage. And of course, students are constantly confused by that because they said voltage can be positive or negative. The positive and negative is just referring to or telling you what the charge on the inside of the membrane is. Is it positive or is it negative on the inside? Okay. So therefore, if I had to say, and of course, the resting membrane potential of a cell can range from these two things here, depending upon the type of cell and the type of membrane. Are we talking about the plasma membrane, or what are we talking about? What is the resting membrane potential across the inner membrane of a healthy mitochondrion? A healthy mitochondrion that's doing oxidative phosphorylation, okay, has one of the highest resting membrane potentials, okay? And this is a high, and you saying, well, that's not a high number, it's a negative number. This is representing a very large difference in charge across the inner membrane of the mitochondria. Okay, this is a large number. Okay, it's not a large negative number, it's a large number, and the negative sign is telling you that the matrix side of the inner membrane is negative compared to the non-matrix side. So if we had a mitochondria, outer membrane and inner membrane, we know that the inside of the membrane is negatively charged and the outside of the inner membrane is positively charged. And the inner membrane of the mitochondria is probably the membrane in biology that has the largest, most powerful, okay, membrane potential. Nice, healthy mitochondria. And what's responsible for that membrane potential in the mitochondria? The electron transport chain. And what is the electron transport chain doing to make the outside of the inner membrane positively charged and the inside negatively charged?